thank you for that generous introduction, and thank you all for coming out this morning. Ever since I was a student, I've been fascinated by the idea that earth sciences and life sciences are not separate universes, and that for many questions of importance in the life sciences, it helps to place events in the context of Earth's physical history. And for many important events in Earth's environmental history, it helps to know what the biological contribution is. And so today I'm going to just talk about something that I've thought about off and on probably for the last 30 years, and that's complex multicellularity. And again, I think this is something where a satisfactory understanding of it will require that we embed it in the world of Earth's physical history. And now this, there we go. So just to give you some prologue of where we're going to go. Again, there is this sense now in many countries of questions of life beyond the Earth. And I think we can say with some confidence that while Earth may not be the only body in our own solar system that has ever supported life, it is certainly the only one that supports complex multicellularity. That said, as many of you will know, in a relative sense, complex multicellular organisms are evolutionary latecomers. The first evidence for complex multicellularity comes along about 85% of the way through the history of our planet. And we can actually examine the phenomenon of complex multicellularity through a number of lenses. There's a large literature from developmental biology and functional biology, but I think that we have to use the fossil record to embed those ideas in the context of Earth's physical history, which, as you'll see, I think is an important component of this. And much of the work in my lab for the last 30 years has really been trying to use what we know about the physiology of organisms to build a bridge between environmental history and biological history. So that's where we're going. Let's just start out by saying, what is a multicellular organism? That sounds very simple, and on the surface it is. A multicellular organism is one where the total of the multicellular aggregate is the unit of selection rather than each individual cell within that aggregate, be a colony. But from there, the various modifiers bring a little complexity to the issue. So multicellular organisms can be clonal or they can be aggregative. Everyone in this room is a clonal multicellular organism. That is, you begin as a single fertilized egg, and all the cells in your body represent successive divisions of that cell. But that's not the only way you can be multicellular. So, for example, if you look in the upper right, those are slime molds, and they're aggregated. They spend most of their life as single-celled amoeba-like organisms, and only in the presence of an environmental signal, usually starvation, do they aggregate and differentiate into a sporangium that produces spores. At the same time, multicellularity can be obligate or facultative. Again, we are obligately multicellular organisms, as are plants. We have to go through a multicellular phase in our life cycle to complete it. Whereas a number of other eukaryotes, including the slime molds in the upper right, are facultative. They will only become multicellular under particular environmental conditions, and the completion of a life cycle does not necessarily depend on that stage of it. And then, where we're going to spend most of the time is differentiating between simple and complex multicellularity. I'll talk about that in some detail, but what you see in the lower two images there are examples of both of those. The one in the middle is a green alga called monostroma. It's a single cell thick just sheet of cells, very little differentiation of cells, very little communication between cells. And as opposed to that, from that nudibranch mollusk that you see in the lower right, animals are complex multicellular organisms with a host of differences between those and the simple ones. So let's talk about simple multicellularity first. A number of examples. It's fairly common within the eukaryotic realm. In these organisms, which are basically usually just filaments, sheets, balls of cells, there may be molecular adhesion between adjacent cells, but that's not ubiquitous within organisms of this type. Importantly, communication between cells and the transfer of resources between cells is limited. Cellular differentiation is limited. And importantly, from a functional point of view, every cell is in direct contact with the external environment, at least during phases of the life cycle where there's nutrient acquisition. So that's a description. And this is found within at least a third of all the major groups of eukaryotes. Very common. Most of these groups have relatively low diversity. So it's in some ways very successful and common in the number of different groups that do it. But within each group, the advent of simple multicellularity has not resulted in a dramatic increase in diversity. Now, just a note to remind us all that simple multicellularity and in fact cell differentiation are not exclusive to eukaryotes. What you see here in the upper left is a sulfide oxidizing bacterium, a simple filament called Vegetoa. On the right in the upper part is Anabena, a cyanobacterium that is not only a simple multicellular organism, but differentiates 3 different kinds of cells. Cell differentiation to a limited extent occurs within the bacteria. And then in the lower right, you see myxobacteria that can actually develop a fairly interesting macroscopic stage of their life cycle. And then just this fossil, which is not quite 2 billion years old, that documents that within the prokaryotes, simple multicellularity has been with us for billions of years. Now, one can ask, what are the functional advantages of being a simple multicellular organism as opposed to being just a simple unicell? I'm a great fan of this work that was done more than 20 years ago, where they took single-celled algae and they could grow them for however many generations you wish. They will always be single-celled. And then they introduced into the cultures predatory protists. And as it says here, within 10 or 20 generations, the algae were obligately multicellular. They were making clusters of anywhere between 18 and 16 cells. And even if you remove the predator at this point, they would continue to function as simple multicellular organisms. The reason for this is pretty clear. That is, the predators can eat single cells. They're not capable of eating these multicellular aggregates. So there was a very strong selection pressure in favor of simple multicellularity. Now, another nice experiment that says something else about how multicellularity can occur. This work from Dayel et al. About a decade ago from the lab of Nicole King at Berkeley in the US, is working with choanoflagellates, sister group of animals. Generally speaking, they are single-celled organisms, but in these experiments, when the choanoflagellates were subjected to the presence of their favored prey bacterium, some molecular signal from that bacterium induced a multicellular stage. And that seems to be something, it's at least conjectured, that may help in prey acquisition. There's actually, if you're interested in this subject, about 6 months ago, there was a very nice review of multicellularity by Tong et al., the laboratory of Will Ratcliffe at Georgia Tech in the US. And as you can see, there are a number of different functional factors that may play a role in the acquisition of multicellularity in different clades of organisms. Certainly, predation avoidance is an important one, maybe the principal one. For things like fungi, efficient utilization of patchy resources is probably important, and some of the others may play a role as well. And I think what's important about these studies is if you ask, how do you go from a single-celled organism to a human being? That's a very difficult issue. Whereas if you can understand that there's a functionality to the early stages of multicellularity where you may have 20 cells, 100 cells, rather than the several trillion that you have in your own body, it shows that there is a functional pathway that can eventually lead to complex multicellularity. I'd also point out another very recent paper by Monforti and Soleil, which explores number 9 in detail. So if you're interested in the subject, I really recommend both of these papers. Okay, so that's simple multicellularity. Now, when I talk about complex multicellularity, what am I talking about? Well, in complex multicellular organisms, there is molecular adhesion of adjacent cells. There is very good communication between adjacent cells. And not only is there communication, but there can be directed communication. So in your body, your cells on their surface have things called gap junctions. In the bodies of plants, they have things called plasmodesmata. And these are channels from which simple nutrients and signaling molecules can pass from one cell to another. And they can be directional in the sense that if there's a cell here and it's surrounded by other cells, it may send a signal only to the cell that is adjacent to it on its right and not to its left. And that opens up the possibility of complex tissues and structures. Pretty clearly, cellular differentiation is shaped by a developmental program that is much of your own genome consists of cells that I would consider middle managers. That is, some cell tells them to do something, they do something in return, and that allows the development of complex three-dimensional structure. And again, a functional complex or a functional consequence of this is that in complex multicellular organisms, only a subset of the cells in the organism are directly exposed to the environment. For most cells in your body, for example, the cells are mostly surrounded by other cells, and that has some real consequences. And in fact, because of this, I've come to think about complex multicellular organisms as organisms in which the constraints of diffusion, whether it be of oxygen, nutrients, or signals, are circumvented by tissues or organs that facilitate bulk transport within the cell. Now, it turns out that unlike simple multicellularity, which has evolved many, many times, there's only about 6 groups of eukaryotes that manifest complex multicellularity. Perhaps a few more depending on how you view the phylogeny of fungi, but there are complex red algae, complex green algae, and their descendants, of course, land plants, complex brown algae, animals, at least 2 and perhaps more groups of fungi. And again, although there are many fewer groups that have complex multicellularity, they have tremendous diversity. It's something like 95% of all described eukaryotes are complex multicellular organisms. Now, if my friend Columbano Vargas from Roscoff were here, he would immediately stop my lecture and say, well, but we haven't just, we don't know what the diversity really is of simple eukaryotes. And in a sense, that's true. And some of you will know the magnificent work of the Tara expedition, which is a largely French undertaking. And that does show, yes, it's probably the case that the described diversity of simple eukaryotes is only about 10% of what's out there. On the other hand, I would say the same thing is, I'm sure, true of insects, is true of nematodes, is true of fungi. So I think that the idea that there is tremendous diversity within a small number of complex multicellular clades is still the case. Just a little aside, one of the questions is, where does this sort of developmental framework, genetic framework that gives rise to complex multicellularity come from? And at least some of that program comes from their unicellular ancestors. One of the things to keep in mind is that multiple cell types within eukaryotes is not an innovation of multicellular organisms. In fact, most single-celled eukaryotes differentiate cell types as part of their life cycle. And some nice work by Middleton and Michaud some years ago showed that simple multicellularity in the green alga ball box has essentially the same genes controlling the differentiation of somatic and reproductive cells within that multicellular structure as we find associated with cell differences in the life cycle of its single-celled relative, Paleontological. So again, the roots of developmental control and complex multicellularity come from single-celled ancestors, although a great deal is also since then. Now, let's look at this problem of diffusion. The cartoon at the top simply shows necessarily what happens as a three-dimensional organism develops. You start with a single cell that's everywhere in contact with the environment, but pretty soon you have, as you see on the upper right, that light gray cell is no longer in contact with the environment at all. It's completely surrounded with other cells. Now, there's been some nice work. This is from a paper that we wrote, which is after a paper by Bruce Gruniger, which is after a paper from about 100 years ago. But what it shows is just the sort of maximum size that a single cell or a single multicellular organism can attain as a function of mass-specific metabolic rate. And what we see here is just empirical data that show the relationship between those two for organisms from a micron to a centimeter in thickness. And what's important in this diagram is that these higher angle lines are simply showing what is possible at different levels of atmospheric oxygen. So even at present day oxygen levels, it's 1 PAL, about the thickest you can be as a single cell or an organism is on the order of a couple of millimeters, maybe to a centimeter, because that represents the limits of diffusion as cells use oxygen. Now, if you're talking about 1% of present day oxygen levels, which as we'll see characterize much of the Proterozoic eon, then we're talking about something that can only be maybe a couple hundred microns in dimension. So what this shows us is that for simple multicellular organisms, the size and complexity that an organism can attain is tied to both the environmental availability of oxygen or nutrients and at the same time to its structure. And this other cartoon here simply makes the point that there will necessarily be a gradient of oxygen, of nutrients, of external environmental signals as you go from the surface to the interior of these organisms. And that's potentially interesting because those gradients are things that induce life cycle changes in single-celled organisms. So it may well be that some of those same relationships to the environment come into play in differentiation of multicellular organisms. And of course, the punchline of all of this is if you can get away from diffusion, then many new avenues for functional differentiation become available. So let's look at our own group, the animals, and say, how do animals get to be so large? I just said that, the largest an organism can be if it's subject to the constraints of diffusion is, less than a centimeter. Everyone in this room is larger than a centimeter. And how do you do it? Well, different animals do it in different ways. Sponges, for example, can be, several meters high. But interestingly, in sponges, with their porous structure, pretty much every cell is in contact with the external environment. So in some ways, sponges are still somewhere along the road to complex multicellularity, still subject to the constraints of diffusion, but able to mitigate them through this porous structure of their body. Now, if we look on the right, where you see a jellyfish, they seem to be thicker, but that's something of at least a physiological illusion. In some ways, a jellyfish is a two-dimensional organism folded bioorganically into three dimensions. So the parts of the cell of the organism that are physiologically active, again, there's at most a few cells between the external environment and the innermost cells. The three-dimensionality, the thickness, comes from the jelly, the mesoglia that is found in the cap of the jellyfish, and that's physiologically inert. So it allows a three-dimensionality without making it subject to the constraints of diffusion. Whereas humans are, I think, truly complex multicellular organisms in that we have tissues and organs that distribute things like oxygen and nutrients through the body. So if you think about how you obtain the oxygen you need for metabolism, in a sense, of course, it goes in through your lungs and is taken up by the bloodstream. And of course, the lungs are, in some ways, a monument to diffusion. They're not where the complex multicellularity comes in the sense of transport. But once the oxygen is bound to hemoglobin, it gets transported to your bloodstream, to your toes and your fingers. And it's really that vascular system that provides really it circumvents diffusion and allows the three-dimensional complexity that we enjoy. Okay, so with that beginning, let's think about this now in a more historical way. And the place I'm going to begin is a little less than 2.5 billion years ago, 2.4 to 2.3 billion years ago, when there is a very important, perhaps the most important single environmental change in the history of the planet. Everything we know about geochemistry suggests that prior to that time, oxygen tensions in the atmosphere and surface ocean were at best very low, basically absent. So for more than the first billion years of life was basically anaerobic. At 2.4 billion years ago, a number of different lines of geochemical evidence tell us that a new world is upon us. One of the earliest things recognized to be telling us about this are what you see in the upper right, that's banded iron formation, largely iron minerals plus silica, the source of most of the iron mined by the human race. And those kinds of sediments are common, and they're basically part of basin development in the Earth's oceans before 2.4 billion years ago, and rare thereafter, except for two moments in the Earth's history where they come back transit. Turns out iron formation cannot in principle form in today's oceans, because when seawater is oxic, that contains O2, iron immediately becomes oxidized and precipitates out. But in the anoxic oceans of the early Earth, ferrous iron can move freely in solution and must have done so to produce these structures. So iron formation is one thing. If you look in the lower right, you're looking at a sand grain from a delta deposited about 2.7 billion years ago in Africa. And the gold color you see in that grain is pyrite. Pyrite is quite influenced by its redox environment. And in the presence of oxygen, pyrite will oxidize to sulfate actually very quickly in terms of your lifetime, the geologic time scale. And so what that grain tells us is that 2.7 billion years ago, that pyrite was exposed on a continent, eroded, carried through a river, and deposited in a delta without ever coming in contact with enough oxygen to oxidize it. It turns out that's broadly the case until 2.4 billion years ago, and then we see it no longer. And there are other redox sensitive minerals, uraninite, siderite, iron carbonate, and they all tell the same story. Before 2.4 billion years ago, they are not routinely getting oxidized in our surface environments. After that, they are. And another line of evidence, I won't go into it in detail, but we'd be happy to discuss it, is sulfur isotopes. Now that we can measure with confidence and accuracy the four stable isotopes of sulfur, we find that there must have been processes on the early Earth whereby sulfur was modulated by photochemistry in the upper atmosphere. And that photochemistry, which is manifest in the total sulfur isotope composition, it's been suggested required oxygen levels about 10 to the -6 of today's levels, so very little oxygen. And as you can see, from a paper by Pascal Philippot, but many people have looked at this, that record is ubiquitous before 2.4 to 2.2 billion years ago, essentially absent after this. So all of these lines of evidence suggest that early in the Proterozoic eon, we begin to have a world that has a permanent presence of oxygen in the atmosphere and surface ocean. And as we see here, here's our hero. And I say our hero because that oxygenation requires life. Specifically, it requires oxygenic photosynthesis done by cyanobacteria. That's the only process that we think is quantitatively able to oxygenate the surface environments. But it probably needs more than that. One of the things that people argue about right now, it's sort of an aside, but it's one that's kind of interesting, I hope. It might be as simple to think that perhaps the Great Oxidation Event just reflects the evolution of aerobic of oxygenic photosynthesis. But there's a lot of reason to think that cyanobacteria existed well before, meaning hundreds of millions of years before with GOE. Nearly all, not quite all, but nearly all molecular clocks suggest that that's the case. And there's at least, it says something like 30 different records where the abundance of redox sensitive elements suggests that there was at least transient local oxygenation of the water column. So if that's the case, then we must have had an earlier evolution of oxygenic photosynthesis that was not in and of itself sufficient to change the world. And one thing that's worth pointing out, and geochemists really think about this, is even today, where you have anoxic waters in the photic zone, and where you therefore have alternative electron donors for photosynthesis, cyanobacteria don't compete very well. It's purple bacteria, green bacteria, things like this that do the photosynthesis. So simply inserting a new physiology into a world that supports anoxygenic photosynthesis is unlikely to affect the world. Now, one thing that I actually like is an idea that comes from the lab of Don Canfield in Denmark. And they suggested that the thing to look at is just the ratio of electron donors other than water to phosphorus. They chose iron because there's reason to believe that on the early Earth, ferrous iron was predominant electron donor for photosynthesis. And if you look at the ratio of iron to phosphorus, if you run out of phosphorus before you run out of iron, cyanobacteria will never do well in their view. Whereas on the other hand, if you run out of iron before you run out of phosphorus, then the only game in town is water as a source of electrons for photosynthesis, and cyanobacteria will thrive. I think that's a really interesting idea because it provides a mechanistic way that we can accommodate an early origin of cyanobacteria with a later oxygenation. We've done some work on this. There's reason to believe that phosphorus availability as a nutrient was limited in Archean oceans. More and more evidence that some of you may be familiar with suggests that for much of the Archean eon, the Earth was still something of a water world. Yes, there were volcanoes that emerged above the water level, at least some small areas of continental crust, but nothing like we see today. And that would result in limited weathering and erosional fluxes. And turns out, interestingly enough, that changes about 2.5 billion years ago. There's a large-scale emergence of continents, which is very clearly seen in the sedimentary record. Why should that happen? It's the hypothesis is that it reflects mantle temperatures. When the mantle is hot, it simply cannot mechanically support thick emergent continents, but as the mantle cools, its ability to support emergent continents exists. Now, at the same time, we can think about remineralization of organic material. That is, if we form cells and then their remains sink down to the seafloor, of course it's happened throughout Earth history, those will be decomposed and release nutrients back into the water column only if there are sufficient oxidants present for bacteria and archaea to do that. And a world without oxygen also turns out to be a world without much sulfate, a world without much nitrate. And as Eva Stoican and her colleagues first suggested, that's a world in which the remineralization efficiency of organic matter is very low. That's still the case, by the way, in lakes today that have very few oxidants. So both of those things would suggest that that the supply of phosphorus as a nutrient would be very much limited relative to today. And then there's a bunch of experimental data, as well as just observations of what happens today in places like the Eastern Baltic Sea, where in an iron-rich ocean, there are a variety of mineralogical ways that phosphorus would be removed from the water column and sequestered into sediments. So in this diagram from a paper that I was fortunate to write with Jihu Hao, a really gifted young geochemist from China, he first of all shows a variety of proposals by different people of how much primary production there was in the Archean relative to today. And you can see that all the proposals are low. You know, at most they're saying 10% of today's levels. Our work basically used two different models for continental emergence, an earlier one and a later one. And it suggested that until you start having continental emergence, that productivity must have been very low. And then as continent emerges, it starts going up. And the punchline then is that if we look at this great oxygenation event, where again, all of these geochemical features tell us that the world is changing, that could at least be the result of decreasing supply of iron to the ocean, because as the mantle cools, hydrothermal fluxes of iron will decrease, and increasing phosphorus availability through the emergence and erosion of continents. So 2.4 billion years ago could at least be the time where we cross over the sort of Canfield ratio, if you will. And after that, cyanobacteria will be ecosystem engineers that will essentially keep alternative oxidants out of the surface ocean in most areas, and it will be a permanent change. Now, I will not swear that that is exactly what happened, but I think it's a reasonable hypothesis. Now, how much oxygen was delivered to the atmosphere and surface oceans at that time? And again, there's pretty much consensus that the answer is not very much. This is a diagram many of you have seen from a review by Tim Lyons and colleagues, and I think it says something very important in kind of a cartoon sense, that the level of oxygen before the Great Oxygenation Event was very low. The level of oxygen since about the Devonian period has been much like today. And between them, we have a long Proterozoic interval of intermediate oxygen. Most people would suggest around 1%, maybe a few percent of present day oxygen levels with largely anoxic oceans below the mixed layer. Again, this is a cartoon, and there's probably more variation in the Proterozoic than it shows. But I think in general, it's telling us something important, that we have this long, low oxygen interval between the early Earth. Now, what can we say about Proterozoic oceans? And I'm going to argue again that not only do they have low oxygen, but low productivity. Again, we're learning almost in real time a lot about the tectonics of the Proterozoic, and they're really interesting and unusual. They're interesting and unusual because although there's evidence of plate tectonically driven mountain building events, there's no evidence that there were actually really large mountains at the time, the Himalayas or the Alps or things like this. And so again, it's probably still the case that although there is more continental emergence than there was in the Archean, that there are limited rates of phosphorus delivery to the oceans. And things like strontium isotope ratios and carbonates would support that. Also, although there's some oxygen, the oxygen supply is still fairly low. So there's now sulfate in the oceans, but not that much. There's now some nitrate in the oceans, but not that much. So again, even in the Proterozoic, it may well be the case that remineralization and therefore the fueling of an upwelling flux of phosphorus was pretty low. And again, as we already talked about, there are reasons to think that an iron-rich subsurface ocean, which appears to be the general state of the Proterozoic oceans, was such that there are a variety of geochemical methods of moving phosphorus from the water column. Nonetheless, there's a new type of cell in town, beginning in the Proterozoic, the eukaryotes. Here are just some fossils from mostly about 1,500 million years ago. It's not just their size that identifies them as Proterozoic, but their structure, and in fact, their ultrastructure seen through TEM. These are problematic organisms. We can't fit them to a particular eukaryotic plane, but they are eukaryotic. And very early on, we start seeing simple multicellularity. There are these things that go back 1,600 million years, just simple sort of centimeter scale, if you will, just blades. There are these crazy things which sort of look like a centimeter scale sausage. And more to the point, in billion-year-old rocks years ago, my former student Nick Butterfield showed that there are simple multicellular red algae. I think that's pretty well established. And another very good student who was in my lab, Shimai Shah, has more recently shown that there are billion-year-old green algae. So early on, we get multicellularity. And in fact, by 1 billion years ago, we have extant photosynthetic groups of eukaryotes having attained simple multicellularity. If you want to see the world record, this is a paper where I've been privileged to work with Lanyan Mao and my colleagues in China. These are simple multicellular eukaryotes. Again, you can see from the size and their organization. If these rocks were Devonian, people would just call these green algae. I think we have to be a little more circumspect, but they are eukaryotic, probably photosynthetic. And what they tell us is that almost as old as the oldest evidence of eukaryotes in general, we see simple multicellularity. So whatever the functional attributes are that favor simple multicellularity, they're there fairly early on. Now, that is in contrast to the earliest evidence for complex multicellularity, which will only happen 1 billion years later. So although simple multicellularity is, I think, probably necessary for the evolution of complex multicellularity, clearly not sufficient. So let's go to the record of complex multicellular organisms. Here's a wonderful place called Mistaken Point in Newfoundland. It's now a World Heritage Site, well dated at 565 million years. And the reason it's a World Heritage Site is that all of those bedding planes that you see exposed in these cliffs are just covered with multicellular organisms. Here you can see some where there's a holdfast, a stipe, and then a more complex layer on it. The question is, what are these? Well, I think it's fair to say that most, if not all of them, are in fact animals, but fairly simple animals for the most part. I think there's evidence that these probably had upper and lower boundary layers or epithelia in biological terms. They only had a very thin surface layer of metabolically active tissues, and it's very likely that each of these, this is sort of a complex of interlocking tubes, and the inside of those tubes probably had either a fluid or metabolically inert material like the mesoglea of jellyfish. There's no evidence of specialized organs for gathering and transporting oxygen, so they must have exchanged gases by diffusion. There's no evidence that they had mouths or digestive systems, so they probably fed by the absorption of organic molecules and phagocytosis by individual cells. There's no evidence of muscular locomotion, so most of them did not move, at least not of their own volition. And organisms like that don't need much oxygen, and their dimensionalities are going to be limited by oxygen availability. Where do they fit into the tree of life? Well, it turns out the description I just gave you of Ediacaran metazoans is very similar to the description I would give of this little organism here, which is called a placozoan. Placozoans are a very obscure group. This is about 1 millimeter across. Only one species described, although genetically they're still settled in the oceans. And again, they have upper and lower epithelia. They have a fluid-filled layer in between. They move by use of cilia, not muscles. They feed by phagocytosis and absorption. They exchange gases by diffusion. So except for their size, these are very similar to what we see in the Ediacaran. Here is a simple phylogeny. Placozoans fill up some of the space between fungi, sponges, and the more complex animals, Cnidarians, things like sea anemones, corals, jellyfish, and Bilaterian, which is most of them diversity with the front, back, top, bottom, left, right. So my guess is that most of what we see in the early Ediacaran animal layer is things that fit in here. So they are macroscopic animals on the road to complex multicellularity, but perhaps not fully complex. Now that said, by at least 555 million years ago, just 10 million years later, we see distributed globally things that are in fact bilaterian. This is Kimberella. It has, you can see, a front, back, left, right, top, bottom. It had a sort of muscular foot that it moved on. You can see in the lower right, we actually have tracks and trails of these with the body fossil at the end of them. It seemed to have viscera that were stacked on top of that foot. If you look in the upper right, it's a beautiful specimen from northern Russia. They seem to have had some device for sort of just combing through the sediments in front of them, much like we would see today in, say, many mollusks, that sort of thing. So this is clearly a bilaterian organism, possibly broadly related to modern mollusks, but clearly not a crown mollusk. And so it shows that we very soon after getting these large macroscopic, fairly simple structures, we begin to get complex ones. And of course, by the time we get to the Cambrian, 15 million years later, we begin to see the diversity of animal complexity that we see today. There are arthropods, there are shell-forming mollusks, there are vertebrates, there are worms, and we have this wonderful record of complex trace fossils that provide independent evidence of functional capabilities. So, as some of you know, and you can infer from this cartoon in the bottom, a lot of geochemical evidence suggests that not only do we see animals appear in the last 30 million years of Precambrian history, but there's the beginning of another environmental revolution, that we begin the path from very low oxygen tensions to ones more like today. This is just a work from Liu et al. 2021. In old age, I'm usually in et al. That's great. These people. This one is based on cerium anomalies, but basically 7 or 8 independent lines of evidence tell us that we begin going toward more modern high levels of oxygen about 600, 550 million years, same time frame as we start seeing large animals. They also tell us, by the way, there are half a dozen lines of evidence now that support the idea that although we start going up to more modern levels of oxygen, this was not the sort of thing where on a Monday there was low oxygen and on Tuesday there was present day levels of oxygen. It's very likely that we didn't reach fully oxygenated oceans until maybe the Devonian period. So this tells how much oxygen. There's a really nice paper, again, from Don Canfield's lab some years ago, where Ben Mills and others did an interesting experiment where they grew sponges at very low oxygen tensions, anywhere from 0.5% to 4% of present day oxygen levels. And they survived, which again is interesting evidence that some animals at least can live under low oxygen tensions. Now, this has sometimes been criticized by sponge biologists because they didn't actually show that these organisms could actually complete a life cycle, but they certainly survived. And if you actually look in ecological surveys of gradients in oxygen in the oceans today, there are animals at extremely low oxygen tensions. Now, it turns out they're usually 100 microns long and 10 microns wide. They're not the kind of thing that's going to show up in the fossil record, but some animals can exist at very low oxygen tensions. That said, the level of oxygen that these people used for their experiment would kill most animals in the oceans today. So rather than simply claiming that animals can live at low oxygen tensions, we have to be more specific. The size of the animal, whether it has diffusion or active transport of oxygen, whether it's locomotion, feeding type, all of those things come into play. And I was fortunate some years ago to have a wonderful postdoc named Eric Sperling. Eric is a professor at Stanford now. And he did this really interesting thing that I, to be honest, never would have thought of. He did a sort of a meta-analysis of ecological surveys along redox gradients in the modern oceans. And what he found, and he looked particularly at polychaete worms, because they actually have a variety of life cycles, or lifestyles. They're abundant, they're diverse. And what he found is that if you had relatively well oxygenated surface layers, there is a lot of both abundance and diversity of carnivorous species. But once you get down to low oxygen levels, and that is, say, below 7.53% of modern levels, you don't have carnivores. And that's kind of interesting, because if we look at the geologic record, carnivores only start coming in right around the end of the Precambrian. And this is, again, it's interesting because years ago, decades ago, it was proposed that carnivory would be an ecological boost to the diversification of animals, as some organisms became specialized as predators and others became specialized as prey and predator avoidance. So Eric's work, and then here's another thing that just comes from a review we did, where if you look at different types of features of animals as a function of oxygen, again, you can see above 8 or 10% of oxygen levels, the biota kind of looks like the normal biota of the oceans. But below that, you have significantly less diversity, the evenness, that is how diversity, how numbers are apportioned among species, falls way down. The number and diversity of predators, carnivores, goes right down. So if we're asking what are the levels of oxygen that make a difference to the functionality of organisms, it's probably not present day levels, but maybe 10% of present day level. And that was clearly exceeded by the time. One more thing happens on this time scale that may help us to stitch all of this together. You're probably tired of hearing me talk about my former postdoc, but this is some nice work by another former postdoc of mine named Duncan Rocks at the Australian National University. And what Duncan did was really a remarkable bit of work. For the better part of a decade, he just sampled shales from through the Neoproterozoic and into the Cambrian and analyzed them to see whether they had biomarker molecules called stigmasteranes that are known to be made by green algae. What he saw is that although there are steranes, and steranes are the geologically stable derivatives of sterols, like cholesterol in your body, there are steranes in 900 to 645 million-year-old rocks, but nothing that suggests that green algae or red algae are important primary producers. At the very beginning of the Ediacaran, 635 to 600 million years ago, there's one sample that showed, I think, ecological importance of green algae in the phytoplankton. And by the middle Ediacaran, that is basically when you start getting animals in the fossil record, every sample shows green algal dominance, and that will continue in the Paleozoic. So interestingly enough, even though photosynthesis in eukaryotes began at least 1 billion years ago, cyanobacteria remain the major primary producers until this time. And then there's a global switch in at least continental shelf environments. So why would that happen? And I think perhaps we get a clue from the distribution of phytoplankton in the modern oceans. You've all seen things like this, where just massive chlorophyll, or basically a substitute for primary production, and therefore nutrients. We have very low production in mid-ocean gyres, high production in coastal environments at high latitudes, and a variety of different models based originally on the work of Penny Chisholm at MIT show that when nutrient levels are low, phytoplankton tend to be very small, right? Mostly cyanobacteria. As you increase nutrient levels, their abundance increases, but only to a certain point. And at that point, larger phytoplankton cells become abundant green algae. And indeed, this continues throughout even larger nutrient levels. You start getting very large phytoplankton, things like diatoms and that. So spatially in the oceans today, there is a relationship between nutrients and cyanobacteria, green algae, diatoms, etc. That is, nutrient level to a first order tells us who's doing the heavy lifting in phytoplankton. And that's, of course, interesting because we see that same pattern in space as well as at time as well as in space. So what may be happening is that the nutrient levels change. And we know that's true for an independent reason. Again, years ago, I was fortunate to work in these rocks. These are phosphorite deposits about 600 million years ago in China. I worked on them originally because they're just amazing paleontological, wonderful stuff. But they're also trying to tell us something about Earth history. There is more phosphate in this formation, the Doshanto formation, by itself than there is in the entire geological record before that time. You can see this from the paper we published 2 years ago, that, on the same time scale as we see more oxygen and more animals, we see a state change in the geological record of phosphates. Now, why might that be? Very quickly, most of you know probably that if you ask where does the phosphate come from that fuels primary production in continental shelf environments today, actually not that much of it comes from weathering and erosion, only a couple percent at best. Most of it comes from remineralization and upwelling. And what we wonder is whether this state change in phosphorite deposition may reflect the real strengthening of the upwelling. In a sense, what's interesting is we do have major mountains forming the Pan-African event on just this time scale. And these seem to be the first time in 2 billion years when we see mountains of the same order as the Himalaya, the Alps today. And so there would be major influx of nutrients. And at that time, and that would more nutrients, more photosynthesis, more oxygen, but then more oxygen means more oxidants for remineralization. And we think you might have something like a positive feedback wherein we would strengthen this this upwelling component, and that of course would help change the world. So maybe, and you can believe it or not, but I think it's an interesting idea, what we're really seeing is something where tectonics is pushing nutrients. Nutrients are producing through primary production more food and oxygen, and that provides the environmental attributes needed for macroscopic animals, and therefore may explain that billion-year gap between simple and complex multicellular. And of course, on the same time scale, we start seeing macroscopic seaweeds and complex red and green algae, which may then result not only from nutrient opposition, but predator. And I won't go into this, but everything I've told you about so far is going on in the oceans. But of course, between about 450 and 400 million years ago, we have a revolution of complex multicellularity on land, where green algae give rise to complex land plants, which of course do have a vascular system that evades the limitations of diffusion. Fungi really evolved tremendously to take advantage of that new food source, the complexity mostly in their reproductive structures, but again, the same time scale. And then of course animals, at least 10 different times, did colonize the land to take advantage. So it all plays out on land. And with that, that'll probably bring us to the end. I hope that you come away with the idea that things like diffusion and the circumvention of diffusion by tissues and organs for bulk transport are important to the functional biology of complex multicellular organisms, and that those are not independent of the oxygen state of the environment. I think at least it's worth considering that nutrient availability is what is linking food, oxygen, and complex multicellularity to planetary history. And then I'll end up, it's something that more and more as I turn gradually into an old man, I think about that. What's interesting about the Earth from an astrobiological point of view may not be that life started here. Life may start in many different places throughout the universe. What might be unusual about this planet is that life has persisted for 4 billion years and therefore been able to evolve in the directions that it has in concert with the physical development of the planet. And with that, I'll just sort of thank the many students and colleagues that I've been fortunate to work with, certainly shaped what I think about it. So thank you, and I'd be happy to address any questions.